Chapter 11, Single Gene Inheritance and Meiosis. By the end of this chapter, you should be able to define allele, genotype, and phenotype. Hopefully you can already do that. You'll also know how meiosis creates gametes, understand how different traits have different inheritance patterns, and learn and understand the genetic basis of human disease through practical applications. Okay, so when you're going through your book chapter, it talks mostly about cystic fibrosis. And cystic fibrosis is a genetic disease. It's caused by a single gene mutation. So there's some change in the nucleotide sequence of the DNA, which creates a new mutant allele, right? And normally what happens is that there's 3 nucleotides that are deleted. So you can see here, in this gene that causes cystic fibrosis, it's referred to as the CFTR gene. It's cystic fibrosis transmembrane regulator protein, I think is what it stands for. Yeah, transmembrane regulator protein. There's another word in there too that it's sometimes referred to as, but the CFTR gene is what codes for that regulator protein. So if you have the mutant version of it, where usually, like I said, 3 nucleotides are deleted, then you have a mutant protein, which means that the protein does not function correctly. Okay, so normally what this protein does is it moves chloride ions in and out of the cell normally, which allows for your mucus to be what it normally is, right? Kind of thin, and it's a healthy coating. In cystic fibrosis, it ends up being really thick and really heavy, and that causes a lot of problems, especially in the lungs, like breathing and other things as well. It also makes a really good breeding ground for bacteria, essentially. So it leads to a lot of infections in the lungs that further damage the lungs, besides being hard to breathe. So it focuses on cystic fibrosis, this genetic disease that this girl has, and she got it even though both of her parents don't have it. So we're going to talk about that, how you can inherit something that your parents don't have as a phenotype, something that they're showing, maybe not physically showing, but it's still a physical trait. Okay? So if that CFTR protein is distorted, then we get abnormally thick mucus buildup here. And like I said, if it has normal protein function, basically chloride ions can go in and out. Water can flow freely in both directions across the membrane so that your mucus is nice and hydrated, it's thin, it's slippery, just like we want it to be. If you have a non-functional CFTR protein, which means that you would have cystic fibrosis, all of those chloride ions get trapped inside the cell. So the water flows into the cell. Remember hypotonic and hypertonic solutions? Now the water's flowing into the cell to dilute those high ion concentrations. So your mucus layer is left dehydrated. It's really thick. It traps a lot of bacteria and inhaled particles. So it becomes a problem for breathing as well as infection. Okay, so now we're going to get into a little bit of the genetics with it. So you might have heard this word before. We are diploid. Diploid just means that we have 2 copies of every chromosome, right? So we have 23 pairs of chromosomes, which means we have 46 chromosomes. So we have 23 pairs of homologous chromosomes, which are just pairs of chromosomes that contain the same genes. So keep in mind, homologous chromosome is different than sister chromatid, okay? Because I think that this can get confusing. So sister chromatid is that you have a replica of a chromosome. So these 2 are the same, right? Homologous chromosomes means you get 1 from mom, 1 from dad, okay? One from mom and 1 from dad, okay? So when you're looking at chromosomes here, again, you have a pair at each of the 23, right? So 1 is from mom, 1 is from dad, and then the 23rd one is your sex chromosome. So you get an X from mom and you get either X or Y from dad, who is the one that determines your sex. Alleles, again, are just different versions of the same gene that have different nucleotide sequences. So even though your parents are giving you their genes, they're passing their genes on to you on these chromosomes, it doesn't mean that they always give you the same combinations. And then you're also pairing it with 1 from mom, 1 from dad. So now you're making up a totally different genetic makeup. So you have different alleles happening than what your mom and dad have. Okay. So these genes that we have that have different alleles are the ones that provide instructions for making proteins. They are physically transmitted from parent to offspring, and then we get a genotype, right? Genotype is a particular genetic or allele makeup of an individual. So this is something you can't see. Again, phenotype is observable or measurable. It doesn't mean you can always see it. So if you get, let's say your phenotype is blue eyes, that's something you can observe really easily. But cystic fibrosis, you can't necessarily see, but you can still observe it. So keep that in mind. If you have something like, say, cystic fibrosis or Huntington's disease, or let's say you have that BRCA1 mutation, all of those things are observable. We know how to observe or measure them, but they're not necessarily something that's really obvious that you can see right when you look at a person. So keep that in mind. That's still a phenotype. Okay. A genotype is the genetic or allele makeup. So let's say that cystic fibrosis, um, we'll go over this in just a second, but let's say the code for it is little a, or lowercase a, little lowercase a. That is your genotype. Okay. So your genotype is going to be letters, basically. It's what your DNA actually is, what your genetic makeup is. And then your phenotype is what is observable or measurable, what you get from that. Keep in mind these A's, this can be any letter. Like we use different letters to do different things. So it could be P, P. This has nothing to do with actual DNA. So you're not looking at nucleotide bases here. Remember our nucleotide bases, those are not involved in your actual gene. They are when you go through like an actual sequencing of your genome, like say you send it to 23andMe, they can see what the nucleotide bases are, like A, T, G, and C. But if you're looking at one gene, you're not going to be using those. You're just going to be using something else for it. We'll get into that in just a little bit. Okay, so we've gone over mitosis. Mitosis is what your body normally goes through for cell division, all somatic cells or body cells. Now we're getting into sexual reproduction, which is meiosis. Okay, so sexual reproduction is a combination of your maternal and paternal alleles. They join during fertilization, and that's what determines your genotype, and it also contributes to your phenotype. Okay. So for sexual reproduction, obviously we've got to have special sex cells. So with humans and lots of other organisms, we have a sperm and we have an egg. These are what we call our gametes. Okay, so sperm and egg, these are our gametes. Gametes are just reproductive cells that carry one chromosome of each, or sorry, one copy of each chromosome. So these guys are haploid. Okay, remember this I know can get a little confusing. We are a diploid organism with haploid gametes or haploid sex cells. And hopefully that makes sense, right? We are a diploid. We have two copies of all of our chromosomes, which means that we're like this complete thing that you've got from partially from mom, partially from dad. But your sex cells, you can't pass on two sex cells, right? You can only pass on one. You've got to have two people to make a baby. So for those two people to make a baby, you're each going to pass on one. So you have haploid sex cells, but you're making a diploid organism. The special cell division that you need to be able to reduce from diploid to haploid is called meiosis. Okay. So again, mitosis is somatic cells, and it's still diploid, always diploid. You're just taking one cell and making it into two, right? And they're identical. Okay. Meiosis has two phases, and we're making it into a haploid cell, so only one chromosome. Okay. So you can see we have mom, she gives up her egg, dad has his sperm, fertilizes the egg, goes through, makes a zygote, which is diploid, has 46 chromosomes now, or 23 pairs of chromosomes, goes through mitosis a bunch of times to make an embryo, keeps going through mitosis as it develops fully into a child. Still has 46 chromosomes, that's not changing. But again, we're going from haploid sex cell all the way down to diploid organism. Okay, so meiosis is only occurring in reproductive cells, so the oocytes and the sperm. Unlike your somatic cells, so your skin cells, red blood cells, muscle cells, anything in your body, reproductive cells do not have a cell cycle. They do have to go through meiosis, but they don't go through it multiple times, right? Girls probably know this even more so, but girls, you're born with all the eggs that you're ever going to have, right? And then you go through puberty and different things happen. But that's why you have to have kids if you're going to have kids at a fairly early age because your eggs age with you, which is not prime for child rearing. Okay, so these do not just keep going through cell cycles and making new ones. They only have one goal, and that's to become haploid. Okay, so for sexual reproduction to happen, again, we've got a haploid sperm. It fertilizes a haploid egg. The result is a diploid zygote. So both of those chromosomes have paired together now, made a diploid zygote. The zygote is going to keep dividing from mitosis till it develops into an embryo and then eventually into a fetus, into a child. Okay, so meiosis actually has the same steps as just mitosis. We go through prophase, metaphase, anaphase, and telophase. But we have two separate division steps. Okay, so we have meiosis 1, which means that we go through prophase 1, metaphase 1, anaphase 1, telophase 1. And for the most part, all the same things are happening. Okay, so here's a few takeaways. You'll notice one that metaphase is different, right? Normally in mitosis, we're lining up in the middle like this. So here we're actually lining up next to each other, okay? So a little bit different. That's the first step that you can tell that you're in meiosis versus mitosis, in case you didn't know that you were looking at a sex cell. But the whole purpose of meiosis 1 is we're going to separate our homologous chromosomes, okay? So through this, we're going to go through metaphase, anaphase, we're going to separate our homologous chromosomes. Now each daughter cell is haploid, okay? So each chromosome still has two sister chromatids. So starting at the beginning of this, prophase 1 are, again, our chromosomes are kind of loosely coiled here. And what they're doing is these homologous chromosomes are getting kind of close together and they're doing something called crossing over, which we'll talk about here in just a second. But that's happening in prophase 1. So I'll go ahead and write that just so you have an idea. Okay, crossing over or recombination or crossing over is happening in prophase 1. Then we go into metaphase, followed by anaphase, and kind of the same things are happening there. Once we get into telophase, now we have our sister chromatids are separated, right? So we have two sister chromatids here and two here. But you can see looking at them, they have like different colors here. That's because of the crossing over that happened here. So unlike mitosis, our sister chromatids here are not identical, okay? So I'm going to abbreviate it SC, not identical, because of that crossing over. Okay, so we've got the pairs of sister chromatids here. So now we're entering into meiosis 2. So we're basically kind of doing the same thing again, but the ultimate goal here is to separate those sister chromatids that we ended up with. So in meiosis 1, we have the 2, right? And then we have the 2 sister chromatids in here. Okay. So now we are going to make that into 4 haploid daughter cells that are then going to develop into egg or sperm. So again, we start with prophase 2. Prophase 2 here isn't quite as robust. There's not as much stuff going on here. There's still, the spindle fibers are forming, they're coiling up, but there's no recombination happening here. So that crossing over is only happening in prophase 1. Okay. Metaphase 2 kind of goes through the same thing. The way it's animated here, I think actually in metaphase 2 it usually lines up the way that you see it in mitosis, but it's not super important for this. You can kind of ignore that. Just remember that it lines up this way in meiosis 1 at least, and that's one of the main differences. And then in anaphase now, instead of pulling apart those homologous chromosomes, we're pulling apart the sister chromatids. Now in telophase 2, we're doing essentially the same thing. We're going through telophase and kind of cytokinesis, dividing it up into those 4 haploid daughter cells that can either develop into an egg or into a sperm. But remember, because of the crossing over and what we call independent assortment that happened in prophase 1, just going to abbreviate it P1, independent assortment plus recombination or crossing over. Okay, because that happened in the first one, you can see that these are all different. So it might be hard, but you can see like the shading on them is different. They all look different, and that's because you're still getting different combinations. So this is why you have brothers and sisters that still don't look identical to you. They look maybe similar, but they might also look pretty different, or you might have very different traits or diseases, just overall genetics because of what happened in prophase 1, which we'll talk about here in a second. So again, no two gametes are identical because of recombination, which we also often call crossing over, and independent assortment. So what exactly are these things that we've been talking about? So crossing over or recombination is just when maternal and paternal chromosomes pair together, they physically exchange DNA segments. So you can see here we have our sister chromatids and a duplicated maternal chromosome, a duplicated paternal chromosome. So these are sister chromatids. They are the exact same, coming from mom, coming from dad. These are non-sister chromatids though, right? We just have one from mom, one from dad. They're gonna go to pretty close together basically. And again, this is happening in prophase 1. And then they're going to cross over to each other to form different chromosomes basically, or chromatids, sorry. So this is what makes them different, is they're actually physically exchanging these DNA segments. You also have something called independent assortment, and that's just that alleles of different genes are distributed independently of one another. So that means that, say your dad has blonde hair, blue eyes, and your mom has brown hair, green eyes. Just because your dad has blonde hair, blue eyes, doesn't mean that he's going to pass on those alleles for both of those things to happen, right? He might pass on one blue allele and one blonde allele, whereas your mom might pass on one brown allele and one, let's say, blue allele, right? It depends on what's dominant or recessive, and again, we'll get into that. But they're independent of each other, so it doesn't mean that you're going to get all the same alleles that your parents have. You are getting alleles from them, but if they have a choice, right, like let's say they have capital A and lowercase a, they might give you capital A here and then give you lowercase a somewhere else. So they're independent of each other, okay? Which means that you can produce 2 to the n distinct gametes. And n is the number of unique chromosomes. So again, in humans, we've got 23 in meiosis, at least in our haploid cells. So 2 to the 23rd power, we've got over 6 million combinations that we can have of unique chromosomes. So there's quite a few, and then you pair that with mom and dad combining, there's a lot of different combinations that you can possibly come up with on top of that were already unique. So you can see here we've got 2 equally probable arrangements of chromosomes at metaphase 1. Then we're jumping down into metaphase 2 and then into the possible combination that we can have in our gametes. So our sex cells down here, either sperm or egg. So you can see like we have mom and dad, let's say, and they're going to go through recombination. And then once they get through here, we've got different combinations. So you can see that we've got 4 completely different combinations of possibilities that can happen. Okay, again, the key difference between mitosis and meiosis is the way the chromosomes uniquely pair and align in meiosis. So again, I think this is happening more so in meiosis 1 or metaphase 1. The first and distinguishing division of meiosis is when you see them line up this way instead of, let's say, vertically all in a line here. Okay. Okay. So, how did the girl in your chapter inherit cystic fibrosis? Both of her parents were normal. They had normal phenotypes. So, how did they pass it on to their offspring? And the answer is that people with the cystic fibrosis don't necessarily have offspring with cystic fibrosis either. So, why does this happen? The reason is because it's caused by a recessive allele. So we have recessive alleles and we have dominant alleles. Recessive alleles we usually indicate by a lowercase letter, so like our lowercase a. Dominant alleles are the uppercase letter, uppercase A. And you'll hear me, I say a lot of times little a, big A. It's just easier for me, less syllables, I guess. I don't know if other people really do that, but try to get used to it. I know my students end up doing it a lot too. I don't know if it's because of me, but I think it's just easier. So little a, big A, okay? A recessive allele is just an allele that affects the phenotype only if the organism has two copies of that, okay? So there's a possibility, since cystic fibrosis is caused by the recessive, we would have to have two little a's in order to show cystic fibrosis. So remember, this is our phenotype, this is our genotype, okay? So in order for us to have cystic fibrosis, we've got to have both little a's. So if we have a big A and a little a, it's masked by that dominant allele, which means that we wouldn't have cystic fibrosis. Okay, and we'll get into looking at these Punnett squares here in just a second. So if we have two different alleles, big A, little a, then our phenotype is normal. We don't have cystic fibrosis, but we call that individual a carrier, okay? And they're a heterozygote. So you can either be a heterozygote or a homozygote. Homozygotes have two identical alleles, and you can either be homozygous dominant, so you have two big A's, again, your phenotype is normal, or you can be homozygous recessive, where your phenotype is that you have cystic fibrosis or you have a mutant phenotype, okay? So heterozygote versus homozygote, and again, homozygotes have two different kinds. Okay, so please keep that in mind. A carrier is only a heterozygote. So say you have a homozygote that is homozygous recessive, they're obviously carrying that little a, but they're not considered a carrier. They're considered a mutant, or they have that phenotype, right? A carrier is only a heterozygote. So be careful with this. You will see it on exams and quizzes. Carriers are only heterozygotes, okay? So you know if someone's a carrier, they've got to have one dominant and one recessive, okay? Okay, so the way that we determine the probabilities of offspring that'll have particular genotypes is we do a Punnett square. And it's based on the genotypes of the parents, and then it matches up all the different possibilities of the gametes that you can produce, okay? And to some extent, you can do this with yourself, with your family, right? But it's not always super easy because a lot of phenotypes are controlled by multiple genes. So like for instance, eye color isn't just on one gene. So you don't just get blue eyes because you got a blue allele from mom and a blue allele from dad. It's contributing on multiple genes. So there's lots of different alleles that come into play. So Punnett squares can get pretty complicated. In this class, we'll usually keep them fairly simple, but let's talk about what they actually are. Okay, so we have dad up here and mom over here. Usually this is how we set it up. So usually we'll have the male over here and the female over here, okay? So you can draw these always on your own, male and female. And then in the case of the girl who has cystic fibrosis, right, both of her parents were normal. So we know that they must have at least been a carrier because affected children or people with cystic fibrosis are recessive, right? Which means that they have to get a little a or a recessive allele from both mom and dad. So both mom and dad must be a carrier for it because that's the only way it can happen. So normally, if there's a dominant allele, you put that first. Okay, so both of them have to be carriers, so we know that they're big A, little a. So this is dad's genotype here. This is mom's genotype here. And now we're going to cross them to see what the odds of their possible gamete combinations are. So we literally just cross them here. Okay, so we have big A going to big A. So our possible genotype here is big A, big A. Now we have big A here going to little a over here. Our possible genotype is a heterozygote, big A, little a, or a carrier. And then we go down here, little a to big A. Again, we have big A, little a. We have a carrier once again. And then we go little a over to little a. And now we have a homozygous recessive individual. So now we can actually calculate out what the probabilities of having these children are. So a lot of times when you're a carrier, you don't necessarily know it, right? Because you're not showing that phenotype. So it's not obvious that you're a carrier necessarily. But if we know that we're a carrier and say we know we're reproducing with another carrier, then we know we've got a 1 in 4 chance of having a homozygous dominant individual. We've got a 2 out of 4 chance of having a heterozygote or a carrier, and we've got a 1 in 4 chance of having a homozygous recessive, okay? So in other words, we've got a 25% chance of having a homozygous dominant, 50% chance of a heterozygous carrier, or a quarter chance of a homozygous recessive. So keep in mind, you can do that. That's your possible genotype probabilities. For our phenotype probability, now it's a little bit different, right? So now we're saying, what's the chance of having a child with cystic fibrosis or a normal? So for normal, we actually have two different possibilities. We can either have a homozygous dominant or a heterozygote. So now we've got 1/4 plus 2/4. So now we've got a 75% chance of having a normal baby and a 25% chance of having a baby with cystic fibrosis. Okay, so some genetic traits are a little bit different, like Huntington's disease is a degenerative neurological disease, polydactyly. There's a few other ones. These are caused by dominant alleles. So these ones are a little bit easier to pass on. So say we have an affected mom, but she's still a heterozygote, so she's got a capital T and a lowercase t, and we have an affected father as well, capital T, lowercase t. So now we do the same thing, right? Big T, big T, big T. We've got a homozygous dominant, big T, homozygous, or sorry, heterozygote, little t, little t. We have, sorry, this should actually be, let's see here. Yeah, this should actually be little t's. Oh, no, sorry, I made a mistake. I was like, how am I wrong? Okay, so little t and big T. Sorry, so this is another heterozygote, and then we've got little t and little t. I just jumped ahead. So now our odds, if we are looking at a dominant disease or any sort of dominant trait, now our odds are 3 in 4 or 75% that we're passing on a dominant allele, which means that we've got a 75% chance of having, let's say, Huntington's disease, and only a 25% chance that your kid would be normal. Okay, so this is how we do Punnett squares. I recommend practicing this a lot. Normally in class we do something with gummy bears, and that's a really easy way to practice this, but there's other resources. I'll try to post them for you guys to be able to practice. Okay. But keep in mind that these can get really complicated, and we'll probably keep them fairly simple. So we just did one that just had 2 alleles, right? You might see some that have 4. Okay. So you will see this, and you basically do the same thing. It does get more complicated, but keep in mind that multiple genes can contribute to phenotypes. And this definitely comes into play with cystic fibrosis as well. Some people have such severe cystic fibrosis that they can barely walk because they can't breathe very well. They die a lot at a lot earlier age. And some are okay, like this girl in the chapter who can actually run, and she had to train herself up to run. But again, it's influenced by multiple genes. So example, this TGF beta 1 influences the immune response to infection. So if you have that, you're able to fight off infection a little bit easier. Okay. So this is an example of a more complicated Punnett square. Again, you just follow the alleles through gamete formation using this Punnett square and still predict the likelihood of offspring homozygous, being homozygous recessive for the disease. Okay, so again, this is just if you have multiple genes contributing to it, which does happen often. A lot of times we will either pretend that it's not happening or we will go with a gene that is only one. But you will still see this from time to time, so make sure you understand it. And it's basically the same thing, right? Big A, little d, little, or sorry, big D, big D. And now we have got big A, big D, little d, big A, little a, big D, big A, little a, big A, or sorry, big D, little d. Okay, so it's just going through the exact same way. Don't get too complicated about it. Just make sure that you follow it along. And for these ones, it will definitely be easier for you to have like a pen and paper nearby to use scratch paper for this. Okay, so how do we treat some of these genetic diseases? One of the main things is we've got to figure out where they are, right? How they're working. But we've got to understand how these modifier genes contribute to the disease, which is how we can kind of figure out what the therapies are going to be. So over time, even in the last like 20, 30 years, we've really increased the life expectancy of cystic fibrosis. It used to only be in their 20s. Now it's getting closer to 40 years old. I think it might even be around 40 now because this is a little bit old. Um, for example, there's something called Calideco, and that's pretty effective at restoring function to the malfunctioning CFTR membrane channel, which is pretty cool. And there's different treatments that they can do. Like the girl in the book wears a vibrating vest that kind of breaks up all the mucus in her lungs. And she also does like a salt therapy treatment. So there's different things that you can do to kind of lessen it to some extent. But it again depends on what alleles you have on the different genes that do contribute to it. Okay, so this actually isn't your class activity. The class activity that's associated with this is more so looking at mitosis versus meiosis. But if you can, I would recommend writing this down, maybe pausing this here and doing this. So we're looking at allele color inheritance patterns or genotypes. Normally we do it with Mike and Ike's. If you wanted to, you could go buy some Mike and Ike's and actually do this, but you don't need to. If you just want an excuse to go buy candy, go ahead and use it. But if we're looking at red, so this is just using another letter here. You could write this out just as capital R, or capital R, G. This gets confusing though. Normally, if I tell you, okay, make up a genotype for cystic fibrosis, you wouldn't use CF, right? You would use maybe C and then little c, or you might use big A, little a, or if it was cystic fibrosis where it's recessive, you'd use little a, right? You don't normally want two different letters because that gets really confusing, especially because sometimes there's multiples, okay? So when we're looking at this, we can see the instructions up here. Red is dominant to yellow, green, and orange. Yellow is dominant to green, orange, and red. Green is dominant to orange, recessive to red and yellow. And then orange is recessive to red, yellow, and green. So orange is basically just recessive. So we know if we have two reds, two R's, then it's going to be red, right? Because reds are most dominant. Again, if we have a red and a green, green is dominant to orange, but it's recessive to red and yellow. So we would still have red here, but we're a carrier for green. Red and yellow, again, it's recessive to red, so we would still have red here, but we're a carrier for yellow. Red here. And go ahead and do this on your own. You can pause me and actually go through this if you want, just to make sure you have the right answers. Green and orange. Orange is recessive to everything, so we would still have green, but be a carrier for orange. And then yellow, and yellow is obviously just going to be yellow. Yellow and green. Green is actually recessive to yellow. Yellow and orange. Orange is still recessive to everything. Green. And then we have orange. Okay. So that's just an example of going through. I actually think it could be a little confusing to you to do this, which is part of the reason why I didn't give you the class activity, but I wanted to explain it here because sometimes you do have to look at these instructions to figure out what's dominant and what's recessive to others. The more likely scenario is I will have you say, let's say we have a mom that is colorblind, and colorblind is also recessive. Okay, so we're going to put her as little b, and we have a dad who's normal but is a carrier. So he'll be big B, little b. Okay, and now we're going to do our crossing over. So big B, little b, and normally it's easier to write the dominant one first if you have it. That way it's easier to see. And we have little b, little b, little b, little b, and little b, little b. Okay, and let's pretend like this isn't color blindness because we'll actually get to color blindness in the next section because color blindness is sex linked, so this isn't actually how you do it. Let's pretend like this is brown hair. Okay, so let's pretend like no, this isn't going to make sense either. Well, we can say this is brown hair. So let's say that big B is black and little b is brown. Okay, so mom has brown hair, dad has black hair. What would our kids have? We've got a 75% chance that they would have brown, and we've got a 25% chance that they would have black in this scenario. Again, not a super great example, but this is more what you're likely to see. Again, there is a combination or a possibility that you'll see ones that have multiple genes. But that is less likely, okay? But be prepared for it, and it's the same thing, right? So you're just doing it in the same way. It just gets complicated because you have a lot of different letters in there. But definitely use some practice, watch some videos, and get really familiar with this because you're going to be seeing quite a bit of it in this chapter. And the next chapter, we're going to get into sex linked, which means that it matters whether it's a male or a female. Okay. So ignore the sex linked stuff for now. We'll get into that in chapter 12. This is just basic inheritance. So we call this inheritance like this, where we write out the genotypes like this. This is simple inheritance. Okay. And then we're going to get into sex linked next time, which is what we call complex inheritance.